Okay. Hi, everybody. I hope more of the clinical faculty will be coming. Gene, you know, that's, we hope. Anyway, I'm delighted to introduce Gerold Bongers. Is that how you pronounce your last name? He is a native of the Netherlands. Did his PhD there, and then he came to do a postdoc with Sergio Lira. We were just talking about a phenotype he stumbled onto that seemed to be related to the microbiome, and it sounds like it transformed your focus. After completing that postdoc, he worked at Janssen Pharmaceutical for a couple of years until he was enticed back to Mount Sinai by Miriam Merad, where he's creating, and he'll tell us about a translational research center in microbiome. This is a really, really important area. Certainly has a major impact on liver disease of all types. We've stumbled into it in our NASH work, and so not just the lab scientists, but also the clinicians should be thinking about this, because there's little doubt that in your careers, if not mine, microbiome therapies will be available to impact at least on bowel and liver disease, and probably a lot of others as well. So thanks, Joe.
Okay.
By the way, I have to leave at 1:30 to catch my plane back to Salt. Gerald knows that I don't Salt.
Yeah, so my main purpose here is to introduce this new center that we're forming. I joined back Sinai after 3 years being in pharma to start this new Microbiome Translational Center. And as Scott alluded, that I was recruited back by Miriam Murad. So my talk has several topics. One is just the center, so what we do, how we can help researchers here at Sinai, and what the services are that we offer. And then I'll go into the work that I've been doing while I was in Sergio's lab. So there are two topics there. One is the role of the microbiome in an intestinal neoplasia. And the other model that I worked on is how the microbiome affects, how it leads to changes in a murine ileitis model. And the last is what I've done in pharma, and that's talking about the development of a therapeutic that is microbiome based, and how you can utilize that to treat, in this case, inflammatory bowel disease. So we'll start to introduce the Microbiome Translational Center. And just a little bit of a highlight on the microbiome. The microbiome is, as already alluded, gets implicated in a lot of different diseases. One of the ones that is where I both work clinically and in pharma on is inflammatory bowel disease, where you can see changes when you do sequencing of the gut microbiome. There are specific changes in the gut microbiome associated with either ulcerative colitis or Crohn's disease. And more importantly, when people treat, when they take the fecal microbiota from a healthy donor and give that to IBD patients, they can see clinical remission. So it's not just that you see changes in the microbiome, you can also use the microbiome to treat disease. So, and that's the combination that we also want to focus on in the center. Similarly, in type 1 diabetes, even before the onset of disease, you can already start seeing, I don't know if you, yes, you can start seeing changes in the microbiota. So here is shown just the overall diversity in the gut microbiome. And people that turn out to develop type 1 diabetes, you can see this diversity being leveled off, while in healthy subjects, the diversity is increasing and with later age. And another very booming field is in immunotherapy, where checkpoint inhibitors are given to certain cancers, and their therapy is responders and non-responders associated with certain microbes that live in the gut. So you can stratify patients that probably will respond to therapy depending if they are high in Clostridialis, at least that's the summary of this paper, and they will not respond well to therapy if they're high in Bacteриalis. And the other reason that I'm here is also because there's this gut-liver interaction that is very strong, and that's why we think it might be important for you guys here to focus on the gut microbiome as well, because of this crosstalk between the gut microbiome and the liver. So what are the things to think about? One is therapy optimization. So for example, as I alluded with immune therapy, you can stratify patients depending who will respond and who won't respond to certain therapies. And that is just one of the examples. The other one is a biomarker. So for example, in type 1 diabetes, you can start to predict who will develop disease. And sometimes you see, and also, for example, inflammatory bowel disease, certain microbes are more abundant in disease subject than others. The other subject is, so that is just who is there and characterizing what type of microbes live in the gut. The other aspect is that you can treat. So I gave the example in IBD where you can give the healthy fecal microbiota of a healthy person, give it to IBD patients, and they go into clinical remission. And the other is that the microbiome in itself, so instead of transplanting a whole fecal microbiota, you can derive certain subcocktails and provide that as a drug. And that's what I worked on in pharma, where we devised a cocktail of a certain set of bacteria and that you can use to treat. So you don't need to do this whole fecal microbiota transplantation. You know exactly what you're giving, you know exactly what the risks are. So then the center is really here to provide a centralized hub for microbiome research. There are already some faculty that do microbiome research, and the idea is for the center to bring all these resources together. And for investigators across Mount Sinai to make it easier to interact with, there's one central talking point. Yes, and the other is to enhance interactions. And then the overarching goal is really to be able to translate the findings that we do either preclinically or from clinical samples and inform medical practice and eventually develop new therapies, whether microbiome based or molecules that are derived from the microbiome. So just for the scientists, the principal investigators here, so the idea that the center helps with consultations for the microbiome projects. So we are there to help to design the either preclinical or clinical microbiome studies, and will help to write the grant. So for if you write your grant application, we'll provide text with the appropriate methodology and appropriate controls, etc., to make it as seamless and as easy as possible for investigators here. So what we have is, for example, SOPs, how best to collect. And there are several ways. If you just want to do shotgun metagenomics, there's one way of collecting. But if you also want to have the ability to do more with the microbes, so going beyond the step of characterizing who is there, but also functionally characterize the microbes themselves, that's another way of collecting the samples. And so far we have SOPs for the stool collection. We also are working on oral collection. I'm not sure if that's applicable for this group. And we're developing others, such as the skin microbiome as well. So far, we're using the Human Immune Monitoring Core for biobanking. So their sample tracking system is used, and they've been using this for a long time. And the other aspect is we will have a specialized nucleotide isolation methods where everything is cryoprocessed. So it's really important. If you want to extract nucleotides from microbes because of the membrane structure, you need specialized equipment and making sure that you crack the membrane. So there's a combination of chemical and mechanical lysis. The additional piece, so once you have the nucleotides, it's all going on to next-gen sequencing. And together with the Clemental lab, we're developing a pipeline where you can seamlessly analyze your data. So it's all web-based. You upload your data, you can play with it yourself. And that gives the basic analysis, all the basic plots for microbiome analysis. And then once you want to modify your plots, you come and talk to us, and we'll work with you to get the plots out that you want to show in either your papers or your grants. So besides that standard, I don't know how familiar you are, the standard CHIME analysis, we're also offering shotgun metagenomics, and that has the advantage is that you can also look at exactly at what type of genes and pathways are there. So that goes a bit beyond the more commonly used 16S RNA analysis. The other piece is, so that is to just characterize what is there. So that is helpful for biomarker analysis. The other piece is to understand what the microbes are doing. So together with the FATE lab, we have anaerobic culture systems available, so where we can take a fecal sample, grow them aerobically and anaerobically, and then plate them in 96-well plates. So you have a complete collection of all the microbes, most of the microbes that live in the gut. And then you can define either sub-communities of those or put back the whole community that you can grow. The advantage is, in that case, you know exactly what you're working with, and you can try to tease out what type of microbe might be influencing your phenotype. This is all automated as robotics, so it's relatively effortless, and we're also going to most likely get more colony pickers, so you can get a bigger depth of colonies, so it's not a manual picking anymore. And then we have a whole pipeline to do strain level resolution. So the other aspects is, so often people characterize their microbes by 16S sequencing, but that doesn't always go to the strain level. So we have a pipeline also to drive complete genome, so you can actually get that strain level identification. And yes, the other aspect is we offer the not abiotic mouse model. So once you have your clinical fecal sample or from any other model, either the whole sample or the aerated collection that are grown on these plates, you can use those to colonize germ-free mice. They will only colonize them with the microbes that you give them, and then you can use these mice to run whatever model is suitable for your disease. So the use cases, for example, that can help with research is there's germ-free control. So that's just addressing the question, do germs matter in your model? So you can use a germ-free mouse for that. The other is to colonize with specific microbes, so you can see whether a certain set of microbes change your phenotype or not. And the other feature, you can use actually clinical samples to colonize these non-abiotic mice. So there are so all these mice are kept in not abiotic isolators. So we have 10 of these germ-free breeder isolators and a couple of smaller ones to do your clinical experiments. And we're also in next year requiring these positive pressure cages. So those allow the mice to stay germ-free for a short period of time, but much better than the current system. And it's not as expensive as running a complete germ-free experiment, because those are usually more costly. So we're also expanding into different areas. So this is mainly focusing on the gut microbiome. We're also developing tools to look at the virome that lives in your gut, as well as metabolomics. And metabolomics is really the question, what type of chemicals these microbes are making and how they are interacting with the host. And then the idea is to, besides doing this shotgun metagenomics, also to integrate these data sets, so whether that's from the clinical or preclinical models, together with the immune phenotyping, so you can get a complete picture of the data set, both from the microbiome side as well from the immune phenotyping side.
Next question.
Yes.
So in your previous slide, so when you transplant the microbiome from mice, so how many percentage of microbiome survive in the mice?
So the models that I used, about 90%, they stick in the mice. It's just the relative abundancy sometimes are slightly different than what you would see in the fecal sample, for example. For certain microbes, they like the mouse environment is slightly better than other microbes. So it's not so bad. And with these aerated collections, it's even better, because most of those, because it's a slightly simpler community, most of them will actually stick. So this is the overview, what this new Microbiome Translational Center can help researchers at Sinai with. One is to provide consultation, help you design your experiments, help to do the grant writing, help to do the paper writing. So you don't have to be an expert. You can work with the center to do all these analyses. The other is the biobanking of clinical samples. So whether you want to use those for just for the sequencing or in not-abiotic experiments or isolate microbes from, we can help with that as well. And the other aspect is to do the computational analysis. And in this case, we're working on a web-based interface that allows you to do the analysis yourself and look at the data and interpret it. And then you can work with us to finalize it and make it the way you want to show your data. And then the other aspect is there's capabilities to do this high throughput microbial isolation. So taking a human fecal sample, isolating all the microbes from them as an individual colonies and then work with those. So you can either test them in autobiotic experiments or work with those in vitro systems to isolate molecules from, etc. And the last aspect is a mouse and autobiotics, where you can use germ-free mouse or mice colonized with specific microbes and look in preclinical models for whatever phenotype you're interested in. So the way it's structured right now, I am the current facility director. I just joined Sinai back in September, and we're working closely with the two faculty directors, Jeremiah Fate, who some of you already are working with, as well as Jose Clemente, that helps with the computational aspect of things. And then there are a couple of other people that help us with the culturing, the autobiotics, as well as the sample pre-processing and the computational analysis. So now I want to transition into what I have been doing in my previous work when I was still at Sinai. So if there are any specific questions about the center or how you can work with us, maybe ask them now, or otherwise we can also do that at the end.
Well, I'll just make a comment, which we were talking about, is that there are probably very few experiments in mice looking at liver that are not going to be influenced by the microbiome.
Yeah.
So as a variable, I think it's something we need to account for.
Yeah.
Probably, as you said, accounts for discrepant results
between
across
facilities.
The field because they're in different mouse houses. Recognize that. But also in human disease. So Gene is here. He has a strong interest in alcoholic hepatitis. I think there's a growing body of literature around clinical implications of microbiome and presentations. It's really something everybody should be thinking about.
Yeah.
And we would obviously come to you with ideas or questions or suggestions.
Yeah, and we'll work with you to work out the details. And there is a push to obtain more and more clinical samples. And the more we start collecting also from different diseases, because we're all running them to the same both isolation as well as computational pipeline, we can start integrating all these different data sets and disease areas as well. So there's some consistency. And the other thing that we also want to do, there's a body of literature on public data sets. So sometimes it already helps if you want to have a quick idea about what might be going on to have easy access to those public data sets as well. That's the overarching idea. And the easiest is just reach out to me, and if needed, I'll pull in either Jeremiah or Jose for specific questions if that's required. So now I'll go into the type of work that I was doing while I was a postdoc here at Sinai. And this is a story about how the microbiome is involved in a preclinical model of intestinal neoplasia. So just to introduce this a little bit, so there's a subtype of colorectal cancers which are called the serrated pathway, which is an alternative pathway. Most cancers are driven by APC or Wnt mutations. This is a subset that accounts about for 15% of the colorectal cancers, and they're driven initiated by either BRAF or KRAS mutations, and they occur in different sites of the gut. So when we started to look at these sections, we looked at, so there are three major subtypes. One is sessile serrated adenomas, which is called SSA here, or traditional serrated adenomas, but there's a third subset that we focused on, which is called the hyperplastic polyps. And initially, when we started this work, we characterized what is present in the gut, what type of mutations, and both in the sessile serrated as well as in the traditional serrated adenomas, there's an overabundance of KRAS and BRAF mutations, as was known already in the literature. But this other subclass of hyperplastic polyps, many of them we did not detect mutations, at least in these two genes. So then we hypothesized, because HbGF is one of those molecules upstream of EGFR signaling, is a ligand of EGFR, maybe some of those are driven by that. And when we looked at the polyps that we obtained in collaboration with Noam Harpis here at Sinai, there's a small subset of hyperplastic polyps that show a high degree of phosphorylation of the EGFR receptor, and they are not they're mutation negative in both cases. So normal gut intestinal tissues, this is human tissue. There's some phosphorylation in the lamina propria of the gut, but you can see in hyperplastic polyps confining where the area becomes serrated. So if you look at this histology, this is not this smooth epithelium lining anymore. This is a serrated phenotype, and there is what we see, this phosphorylation of EGFR. So we wanted to see if we could model this, in this case in a preclinical model. So what we did is transgenically overexpress HBGF, which is the ligand of EGFR, and we also, to promote cleavage, because this is a ligand that needs to be cleaved with metalloproteases, we couple that to expression of a constitutively active G-protein coupled receptor that leads to cleavage of this and would phosphorylate, activate the receptor and lead to phosphorylation. And in vitro, we can see exactly that when we just express the G-protein coupled receptor, there's no cleavage of HBGF. When we express the membrane, the HBGF by itself, there's a slight amount of constitutive cleavage, but we can promote that by co-expression of the GPCR. So that is all interesting, but when we did this in vivo, what we saw is the development of these serrated lesions in the cecal-colonic junction. So this is the colon, this is the cecal pouch in mice, and then the ileum would intersect here. And exactly at this region, always in the same region, we saw these large masses growing. And histologically, they look like the human hyperplastic polyps. So this is the lesions that we see in the mice. Again, you can see the serrated crypts appearing. And if you compare that to the human hyperplastic polyps, the histology looks very, very similar. And to compare this to what is known in humans, to see if this EGFR signaling was important, we looked at two downstream, well, we both blocked MEK as well as EGFR, and in both cases we can block the formation of these polyps, both in size as well as incidence, suggesting that this EGFR MAP kinase pathway is involved in this model, although we don't have BRAF or KRAS mutations. But the interesting piece is that these lesions always happen in this region. And when we started to look by RNA-seq, we looked at the adjacent region and compared that to the serrated polyp. The type of transcripts that we see, many of them, so besides being enriched in MAP kinase signaling, which is what is known about some of these lesions, we also saw a lot of terms associated with response to bacteria. And one of the most upregulated genes is a gene called RAC3 beta, which is one of these antimicrobial genes. And when we look by staining, for example, so this is the normal gut, in that region, RAC3 beta is normally barely expressed. But in the serrated lesions, many of the crypts start to express this antimicrobial protein. That suggested to us that maybe because of all these transcripts being regulated, maybe the microbiome is important. So to test that, we first put mice on a broad spectrum set of antibiotics. We treated for 15 or 35 weeks of age, and what you can see that the biomass, so the number of 16S copies, 16S is a gene specific for microbes, they start to decrease after antibiotic treatment. But strikingly, the phenotype completely disappeared. So both at 15 and 35 weeks of age, these polyps do not develop. What we see is an enlargement of the cecum, which is a known phenomenon if you long-term treat mice with antibiotics or germ-free rederive them. But these serrated lesions did not appear at all, suggesting that the microbiome is important and somehow a driver for this phenotype, at least in this model. Similarly, when we rederive the mice into a germ-free status, so there are no microbes, it's not antibiotic dependent, so there are no microbes whatsoever present. Again, you see this enlargement of the cecum, but these lesions do not appear, so there's no lesions whatsoever. And even more interestingly, when we just rederive the mice, so in this case, we take the eggs that contain both of the genes, we put them in pseudo-pregnant moms and let them be born from a different mother that has a different microbiome. So we're changing the microbiome from birth on compared to what we used to have. And then we keep them separately so they maintain that microbiome. And also in that case, at 12 weeks nor at 17 weeks of age, there's no development of these type of hyperplastic polyps, while only at 25 weeks we see a small subset that start to develop the disease. But this rederivation, in contrast to antibiotic treatment or germ-free rederivation, there are still microbes present. And if you look at the diversity of the microbiome, so the type of bacteria and how diverse they are still present in normal amounts. So if you compare the mice that develop serrated regions compared to the rederived and then the taconic mothers, they all have the same type of diversity. So it's not that they don't have microbes, it's just different. And if we then look at, principal coordinate analysis, you can see that these mice have a different microbiome. They separate out away. So the re-derived mice, they separate out. They're somewhere in the middle between their taconic mothers and the mice that develop these lesions. So that allows us to specifically look at what is different. So when we're comparing mice that develop serrated lesions compared to wild type, they have a similar microbiome, but the re-derived mice, certain taxa are just missing. Certain microbes are not present. And those, if you look also at antibiotics, they're also hardly present in the antibiotic-treated mice. And those are the type of microbes that we think might be important. So, so when I was still here, we started to follow up this. So using the same thing I just described, taking the fecal material, isolating microbes out of them, and then look which microbe might be important in the phenotype. That work is still ongoing. But that's one of the things that we started to do, because by the sequencing, you don't really get to this strain level. If you do 16S sequencing, often you can already see that the genus phenotype, the genus classification is missing because you don't have the right resolution. So the way to figure out how microbes are really affecting your phenotype is to take them out, culture them, and stick them back in the mice and see if they promote the phenotype. Unfortunately, this is a very long-term model, so these are not very easy experiments because we have to wait at least 3 to 4 months just to see the phenotype. So this will take a while to get to that, what type of microbe is important. The other aspect that we see is that these microbes start to infiltrate the lamina propria. So this is a normal gut, and your microbes are kept outside. The gut epithelial forms a barrier as well as a mucin layer to keep the microbes in the lumen, not to let them infiltrate into the lamina propria. But in these serrated lesions, we can start seeing, so in this case, this is a bacterial probe. This is in situ hybridization, where you can start seeing microbes that are living in the gut. And they're also, and we'll come to that back later, they start to recruit a neutrophil reaction. So in yellow here, these neutrophils are recruited towards the infiltrating microbes. And another way to show that the barrier is not as intact in these type of lesions and as in normal mice is to do this sulfobiotin staining. So this protein will label all surface proteins. So what we do is we take the cecum, incubate this with this NSH labeled biotin, and it will only label the surface proteins. And that's what you see in a normal cecum. You see this red layer outside of the cells, and there's no staining inside of the cell, indicating that the barrier is intact. This biotin compound cannot integrate because the tight junctions are keeping the molecule out. But only when the barrier is not intact, and that's what we see in serrated lesions, this biotin labeling can occur in the lamina propria, suggesting that the barrier in these lesions is not as tight and as functional as in a normal gut, and that maybe explains why we see these microbes infiltrating. The other thing that we looked at is how the inflammatory cells are responding. So we did a microarray comparing the adjacent tissue with the serrated lesions, and we looked at what type of genes are overexpressed. And one of the genes that are mostly upregulated are these S100 genes. And again, when we look at the serrated lesions, we can see that these S100 cells, they're close to where the lesions are, and they're close also where the microbes are infiltrating. And when we do this more accurately by flow analysis, we can see that and compare both to wild type as well as adjacent tissue, as there's an increase in the lesions of these LO6C, LO6G positive, LO6C positive cells. And we also looked at GO1 and cyclic F negative cells, and both these markers suggest these are infiltrating neutrophils. So the next question that we asked is, are these cells actually important for the phenotype? So what we did is we treated these mice for long term with an LY6G antibody, and what we can see is that both that we can reduce the number of infiltrating neutrophils, but importantly we can also reduce the size of the polyp, suggesting that these neutrophils somehow contribute to the phenotype. So in summary for this subpart, what we propose is that it's both, even if you have a susceptible microbiota, so the microbiota that we have normally in the mouse facility here at Sinai, they contain the microbes that we think promote disease, but they don't do that unless you have the right genetic components. In this case, slight increase in EGFR signaling. But even if you have the right genetics, only you can start seeing disease when you have this combination of both the genetic change as well as the microbial change. And that in this case then promotes an inflammatory response that further contributes to this positive forward loop that increases the formation of these serrated lesions. And then I want to switch gears. So just before I left, I also worked on a murine model of ileitis. So in this case, so this is a model of inflammatory bowel disease. And also in inflammatory bowel disease, it's thought that it's both a combination of environmental factors as well as the genetic susceptibility that is contributing to disease. So the genetics doesn't explain the full extent of the disease, but as I already explained in the introduction to there are certain microbiome signals that are seen in disease, and certain microbes are overrepresented while others are underrepresented in IBD subjects. So to look at this preclinically, we utilized a mouse model where the ARH element from the TNF gene is deleted, which will make the transcript slightly more stable, which leads to a slightly increase in TNF expression, and that's what we see here. So while the mice are aging, there's a slight increase in the amount of TNF being produced. And TNF is one of the molecules that is critically important for inflammatory bowel disease, because the major treatment of biologics is all targeted against TNF, and slight overexpression also leads to what looks like a ileitis. So if you compare wild-type mouse, there's very little inflammation, but over time in these mice that lack this AR-rich element, you start seeing an inflammatory response in the gut. They also develop arthritis, which I'll come back later as well. So in this model, we started to look at the microbiome, and what we saw was that, so when the mice are born, so this is a pie chart of the different phyla that are present in the gut. When the mice are born, they sort of look the same. There's not a major difference. But over time, you start seeing an expansion of the firmicutes, in this case, in this model, and a contraction of Bacteroides. Associated with that, and that's also a phenomenon that's seen in human disease, there's a decrease in the overall diversity. So similar to the type 1 diabetes story, also in IBD, the diversity of your microbiome starts to decrease with ongoing disease. And if you look at the multidimensional scaling plot, you can start seeing that the mice that develop this inflammatory response, they all start to cluster together and they start to separate from the wild-type mice. Again, indicating that the microbiome is changing over time. And again, similarly, we wanted to look if there's a specific microbe that is, in this case, associated with disease. So we looked at all the different operational taxonomic units. So that is a proxy for the type of species that live in the gut. It's dependent a bit on the sequence, so you cannot just get to the strain level. So usually you talk about operational taxonomic unit, which just means a certain stretch of the 16S RNA that is overrepresented. But they classify to certain species, and two of the OTUs that we see increase, they all belong. So they belong to a similar species, and there's one Clostridia candidatus or emertis. It's also known as SFB, that starts to increase in these mice, and that's specifically increasing over time. So this is different time points, and this specific strain is increasing while another strain is decreasing. And here it is the same data shown as in a box plot. So at 4 weeks of age, in both the wild type and the TNF-DAIR mice, there's very little of this microbe present, but over time, also in the wild type, slightly increases, but it dramatically increases in the mice that start to develop disease. So here we ask the same question: if we change the microbiome, and in this case we just looked at antibiotic treatment. Can we prevent disease? So in this case, instead of treating the mice when they're adult, they were put on broad-spectrum antibiotics since birth, or they were treated with vancomycin, but in both cases, the ileitis disappears. And this is just one example. So this is the histologies, again, the laminophobia. There's massive inflammation at 20 weeks of age in this model, but the mice treated with antibiotics have no disease in the gut. While, as I alluded to before, the mice also develop an arthritic phenotype, and arthritis is not affected whatsoever. So in both cases, they still develop arthritis in the joints, but there's no gut phenotype. So the gut phenotype is completely microbiome dependent, while the arthritis is not. And this is just looking at the staining again, showing the same data. So in this case, is CD45 staining, just highlighting all the inflammatory cells in the gut. There's a massive inflammation at 20 weeks of age, and after antibiotic treatment, the gut essentially looks normal. So just to summarize this part, so what we see is that with increasing amounts of TNF, there's a gradual increase in the expression of certain antimicrobials. This is associated with a decrease in certain species, and this dysbiosis, this change in the microbiome, it mimics a little bit what is seen in human disease as well. And the inflammation that is seen is completely dependent on the presence of microbes. So this is what the work that I have been doing here while I was in Sinai, and then I joined pharma to work on how to treat disease. So what I worked on is the development of a biotherapeutic. So these are microbes derived from a healthy human subjects, and to see if that can help to treat inflammatory bowel disease. So again, just to highlight, so if you look at the human disease, certain microbes are increased. So, and this is an example for Crohn's disease, and certain microbes are decreased. So if you look at the type of bacteria that are decreased, most of them belong somewhere to the order of the Clostridia order. And the general concept is that there's a dysbiosis in the gut, that the good bacteria start to decrease, the bad bacteria, putative bad bacteria, are increasing in their relative abundance. And then maybe intervening with this might keep people in a healthy state. And as I already showed you in the beginning, there are two clinical studies that test this concept, where they take the fecal microbiota from a healthy person in a very intensive treatment, give it to patients with inflammatory bowel disease, and they can induce, in both these studies, they can induce a clinical remission, suggesting that actual changing of the microbiome is a good therapeutic approach. So what I worked on there is a set of bacteria that were originally described by Kenya Honda, and what they have done in his lab is take the chloroform treated feces, and what chloroform does, enrich for spore-forming bacteria, which are resilient bacteria that have, all the work has shown that those are usually associated with induction of regulatory T cells. So to promote enrichment of those, they chloroform treated them and passes them through germ-free mouse, which basically is selection for microbes that can colonize well in mice. So they enrich for that, and then they looked at 16S sequencing and see which type of organisms are present. And what they come to after this selection procedure, they come to a mixture initially of 23 microbes, but then looking at the sequencing, they subsetted this to a set of 17 strains that are uniquely there. So some of these that are present in the 23 mix, they look highly similar. So they just looked the ones that are most divergent. And they looked at those 17 strains and then gave them again, in this case, to germ-free mice. And what they saw that only when they treat with this mixture of 17 strains, they can induce FOXP3 positive CD4 cells, which are regulatory T cells. Subsets of these 17 mix do not induce, or not to the same extent at least, regulatory T cells, suggesting that you need this combination of multiple bacteria to get the full response. And importantly, when they use, in this case, is another model for inflammatory bowel disease, where you treat with TNBS, the mice get inflammation, and when they treat with these mixture of 17 strains, they can prevent disease. And in this case, the readout is survival. but also when you look at histology, there's a decrease in histology score, suggesting that the induction of these regulatory T cells or through some other mechanism, you can prevent disease, at least preclinically. So this is how it started. So initially, we did a very simple experiment, show the induction that compared to vehicle-treated mice and comparing vehicle-treated or 70 mg treated mice, you can induce these regulatory T cells, but we also looked at how well these type of microbes engraft, as well as a metabolic characterization. So that's initially how I started there. So this is the engraftment. So this is a germ-free mouse. There's obviously no microbes there, so there's no detection of any of the microbes. But when you look at these different bugs, all of them engraft in the mice. So going to your point, if you go with a specific set, and in this case, they're also slightly enriched for bugs that engraft really well in mice, most of the bugs will engraft, especially in a germ-free status. If you look at a full microbiome, some bugs just have a harder time. So not all things will engraft. But you also see major differences in the relative abundances. Some microbes are really doing really well in a mouse gut and others don't. And similarity to the published data, we can see an induction of these regulatory T cells. And what we also did is to look at what type of metabolites are being produced. So short-chain fatty acids are often associated with induction of regulatory T cells. And when you compare vehicle-treated, so in this case, an essence germ-free mouse, for example, one of these short-chain fatty acids, butyrate, is massively upregulated. So it's below detection. So in gray is below detection. There's no detection in vehicle-treated mice, but you can rather easily detect it in mice treated with these 17 strains of Clostridia. But beyond that, there's a lot of other metabolites also changing. And this is even scratching the surface. We don't really understand all the type of molecules that microbes can make. So this is the ones described in the literature, but there's a whole lot more. So to show that this also works preclinically, we used a slightly different model. So in this case, the model is a knockout for IL-10. These mice, when they're kept germ-free, do not develop disease, similarly to the TNF-DARE mice that I described previously. But when you give a microbes to these mice, they start to develop colitis. So what we did is take a human fecal material from, in this case, a healthy donor, give it to the IL-10 mice, so colonize all the microbes from this donor, wait for 2 weeks, and slowly these mice start to develop colitis. And after 2 weeks, we started treating either with a vehicle control or a mixture of these 17 strains, and then look for disease and metagenomic markers. So interestingly, what we can see if you compare, if you compare the vehicle-treated mice to the knockout mice, so these mice start to develop colitis, we see also an increase in, when you treat with these mixtures, you start to see an increase in overall diversity of the microbiome. And as I alluded in the beginning, in one of the hallmarks of inflammatory bowel disease is a decrease in the alpha diversity. And in this case, when we treat with this mixture of bacteria, we start seeing an increase in this, which is suggestive that it might be good. What we also see is the engraftment. So in all these cases, these are associated with the cocktail that we're giving. So they belong to different taxa, and they belong to different taxa. And what we see, for example, if you don't treat, we don't detect it at all in this case. And after treatment, all these things that we're giving, we can actually detect them. So in this case, we're not doing 16S sequencing, we actually do shotgun sequencing. Otherwise, we cannot discriminate from what is already there to what we are giving. So this is a different type of looking at the microbiome. And interestingly, what we also see that certain microbes that are increasing with disease, so in this case, these Enterobacteriaceae, they increase when the mice develop disease. We can bring those down again after treatment. And importantly, we also bring down the histology score. So if you compare a wild type mouse, there's no inflammation. The knockout mice, in this case, after 4 weeks of age, they start to develop inflammation in the lamina propria, but after treatment with these mixtures, the disease is significantly decreased. So this is all the preclinical work. I cannot show other parts of this type of things, but also was involved in the biomarker analysis. And yeah, that's more complicated, but this is the only piece that I can share. So what we've seen in this work, we can give these mixtures of microbes, then graft the mice, even in the presence of a human fecal material. And there's the production of putative beneficial metabolites, including short-chain fatty acids and bile acids. And treatment with these strains prevents disease, at least in a preclinical model. And the question now is, does it work like that also in a human setting? That's what I had to share. There any no questions? Okay.
So, so when you transplant the human fecal to any not only the bacteria but also some other living like so the development of the disease, what's the role of those
it's not well understood. So it's suggested that, for example, short-chain fatty acids, they promote directly through interaction with G protein coupled receptors, induction of regulatory T cells. Same for bile acids, they can shape the milieu, but also they act on host receptors. But that is just scratching the surface, because there are also a lot of other microbes that are not well understood. So Louis Cohen here at Sinai, for example, has also published a study where there's a new class of chemicals that mimic, so the microbes make molecules that look like the human molecules, and they can act both on the same receptors. But the amount of chemical dark matter, let's say, that the microbes are making is massive. We don't understand most of the chemicals, so that's yeah, there's a lot to learn there. And there are ways to characterize this. You can do like this global metabolomics, but even there you have to know what you're looking for. And a lot of the things we still don't know. Except for, yeah, well-known things like short-chain fatty acids that people have looked at extensively. They're released free of microbes, of bacteria, because all the mice are checked by both 16S qPCR as well as culturing, and they no, they don't. But yeah, and they live, but their immune system, for example, is slightly, it's not completely normal, because there's other work that shows that you need microbes in early life to educate your immune system so it gets educated properly. So some certain cells are just absent. Yeah. So definitely this early life exposure is an important event, and obviously germ-free mice don't have that. And even after you colonize them, you can see a lot of the immune cells and suddenly come up. So a lot of microbes will induce regulatory T cells, or they will induce Th17 cells. So how informative is that? It's still, you can learn a lot, but you have to take into account some of those caveats. Because certain subsets, you need that early life event to capture that. But other immune regulatory functions you can readily assess. But you have to be aware of the limitations. And like any model, there are limitations to it. Project funding experiments, does the center also support or give an estimate? Should that all be PI funded for certain setup of certain assays. We'll probably initially start setting it up, but the actual experiments have to be PI funded. That's the plan. So we can sit down and just show you, okay, these are the surfaces, and depending on What you want to do exactly, we'll give an estimate of how much that will cost. Because sometimes, let's say for the sequencing, it depends if you want to use us for sample storage or not. Some investigators already started talking to they want to do the DNA isolation, so then we don't charge for that. But whatever service, well, yeah, we discussed that. So we have a budget for each of the services. So. Sorry?
In your biobank?
Yeah.
So there is history, like how they treat.
So in this case, so what we have banked already is mainly clinical samples from inflammatory bowel disease. And there is a questionnaire associated when they do the sample collection. Yeah, so let's say any of you wants to do a clinical experiment, there are a couple of type of questions that would be nice to have if you want to do a microbiome study, including antibiotic usage, smoking, dietary things, if at all possible, because there are some covariants that are associated with changes, and it's good to capture those, so it helps the statistical analysis in the end. So yeah, and in those cases, for the studies that are already ongoing, we try to include as many as possible of those type of questions.
How long the job remains? How many months? Just curious.
The ones that I don't know exactly. The ones I work with at least a year. Yeah, but they live besides having like a massive secant that at some point becomes problematic for some of them. Some that's not that problematic. I don't know their actual lifespan, to be honest, but for sure 1 year, 2 years. So, but some prematurely died mainly because of obstruction of the gut and malabsorption, probably near the end. So.
Thank you.
Okay, thank you.